Hello everyone. Imagine you have a mixture containing several different chemicals. They are all together in one tiny vial. What you need to know what compounds are present, how much is present, and whether the sample meets its specification. This is where gas chromatography or GC comes into the picture. But what actually happens inside a GC? Today we are going to follow one sample from the moment it enters the injector until it finally appears as peaks on the chromatogram. What is gas chromatography? Gas chromatography is an analytical separation technique used mainly for compounds that can be vaporized without decomposition and that are sufficiently volatile and thermally stable. In simple words, GC takes a mixture of compounds, separates them inside a chromatographic column, detects them individually, and produces a chromatogram. The GC instrument. Before we follow the sample, let's understand the main parts of the instrument. First, we have the carrier gas system. Second, the injector, where the sample enters and is vaporized. Third, the chromatographic column, where separation occurs. Fourth, the oven, which controls column temperature. Fifth, the detector, which detects compounds as they leave the column. And finally, the data system, which converts detector signals into a chromatogram. Carrier gas. Let's start with the carrier gas. The carrier gas acts as the mobile phase of GC. Common carrier gas includes helium, hydrogen, and nitrogen. The carrier gas must be sufficiently pure because contaminants can produce background signals, interfere with analysis, or affect detector performance. The gas flows continuously through the GC system and carries the vaporized sample through the column. Sample injection. Now the actual analysis begins. A small amount of sample is introduced into the GC inlet in a conventional injection system. The inlet is maintained at an elevated temperature so that the volatile components of the sample can vaporize rapidly. The injection technique and inlet conditions are extremely important because poor injection can cause problems such as discrimination, poor peak shape, carryover, or inconsistent response. Split versus splitless injection. In split injection, only a portion of the vaporized sample enters the column while the remaining portion exits through the split vent. This is useful when the sample concentration is relatively high. Splitless mode. In splitless injection, the sample is transferred predominantly into the column, making this mode useful when greater sensitivity is required for lower concentration analytes. The most important part, the column. Now we reach the heart of GC, the chromatographic column. This is where the actual separation takes place. A typical capillary GC column contains a stationary phase coated on the inner surface of the column. As the compound travel through the column, they rapidly interact with the stationary phase while being carried forward by the mobile phase. Different compounds have different interaction with the stationary phase and different volatilities. Therefore, they do not travel through the column at exactly the same speed. One compound may move relatively quickly while another may spend more time interacting with the stationary phase. Eventually, compounds that entered the column together become separated. Why do compounds separate? So why do compounds separate? The separation depends on the several factors including the compound's volatility and their interaction with the stationary phase. In simplified terms, compounds that spend less time interacting with the stationary phase generally move through the column faster. Compounds that interact more strongly generally take longer to reach the detector. The role of the oven. The GC column is placed into a temperature controlled oven. The oven temperature can remain constant throughout the analysis. This is called isothermal operation. Alternatively, the temperature can increase according to a programmed temperature ramp. This is called temperature programming. Temperature programming is particularly useful when the sample contains compounds with the significantly different volatilities. Compounds reach the detector. After separation, the compounds leave the column one after another and enter the detector. The detector responds to the compounds as they pass through it and converts their presence into an electrical signal. Different detectors are selected depending on the analytical requirement. Some commonly used detectors include FID, TCD, ECD, and mass spectrometric detectors. One of the most widely used GC detectors is the flame ionization detector or FID. In FID, organic compounds reaching the detector are burned into a hydrogen air flame, producing ions. The resulting electrical current is measured and converted into a detector response. FID is widely used because of its sensitivity to many organic compounds and its broad application in routine GC analysis. The chromatogram appears. And now comes the result we actually see, the chromatogram. Each separated component can produce a peak. The horizontal axis represents time, usually retention time. The vertical axis represents the detector response. The position of the peak tells us when the compound reaches the detector, while the peak response can be used for quantitative analysis according to the analytical method. What is retention time? The time between injection and the peak's maximum response is called the retention time. Under controlled analytical conditions, retention time can be compared with that of a suitable reference standard to help identify a compound. How quantification works? GC is not only about identifying compounds, it can also be used for quantitative analysis. The detector produces a response for the analyte, commonly evaluated using peak area. In a quantitative method, the sample response is compared with an appropriate reference standard, internal standard, calibration curve, or other calculation approach specified by the validated analytical procedure. System suitability. But before accepting analytical result, we need to make sure the chromatographic system is performing properly. This is where system suitability comes in. Depending on the analytical method, parameters can include retention time, resolution, theoretical plates, tailing factor, and repeatability of response such as percentage, RSD, or relative standard deviation. What can go wrong? Problem 1, ghost या carryover. Problem 2, peak tailing. Problem 3, retention time shift. Problem 4, low response. Summary. So let's follow the complete journey one last time. The carrier gas provides the mobile phase. The sample enters the heated injector and vaporizes. The carrier gas transports the vaporized sample into the column. The compounds separate based on their different chromatographic behavior. The oven controls the column temperature. The separated compounds reach the detector at different times. The detectors convert their response into an electrical signal. And finally, the software converts those signals into the chromatogram be used for identification and quantification. That is the complete journey of a sample through gas chromatography. If you found this video helpful, don't forget to like, share and subscribe.